We often think of cancer as one disease, but cancer in children is a profoundly different beast than cancer in adults. And to really get our heads around it, we have to go deep. Right into the source code of life itself. So let's dive into this incredible new analysis that's starting to pull back the curtain on the unique genetics of pediatric cancer.
First, let's just get a sense of the scale we're talking about. Every single year, around the world, about 400,000 children and adolescents get diagnosed with cancer. It's actually the leading cause of death from disease for this age group, which is exactly why figuring this out is so incredibly urgent.
And yeah, that brings us right to the heart of it. Why is it so different? It's not just that the patients are younger. These cancers are fundamentally different on a molecular level. So what's really going on under the hood?
Well, the answer, as this new research makes crystal clear, is buried deep inside our DNA. It's a totally unique genetic puzzle, and scientists are, for the first time, really starting to put the pieces together.
Okay, so this slide really gets to a key difference. For adults, cancers are often the result of, a lifetime of things. Sun exposure, smoking, just bad luck piling up. And that leads to a ton of genetic mutations. But with kids, it's a whole different story. Their cancers usually come from errors that happen while they're growing so rapidly. And here's the kicker. They have way, way fewer mutations. That means every single one that's there is incredibly important.
So because there are fewer mutations, scientists can actually zero in on the ones that are really driving the train. This whole study was basically a massive genetic detective story, a huge deep dive into the data to find the specific genes that are causing all the trouble.
And when I say massive, I mean it. The mission here was to analyze the genetics from, get this, over 6,500 pediatric tumor samples, pulled together from 13 different studies. They were just looking for patterns, for the genetic bad guys that kept showing up over and over again.
And this is how they did it. Now, I know this looks super complicated, right? But just think of it as a really sophisticated high-tech map. It's a special pipeline they built to sift through mountains of genetic data and pinpoint exactly which genes were the problem children. We can actually break that whole complex process down into 4 pretty simple steps. First, they gathered all that data. Then they had to filter it way down, from over 16,000 mutated genes to just 827 that kept popping up. Next, they sorted those into 4 tiers, kind of like a most wanted list. And finally, they figured out how these broken genes were messing up the communication systems inside the cells.
And that process, well, it gave them exactly that, a genetic most wanted list. For the first time, they could see with stunning clarity which genes were the main culprits, the top dogs driving pediatric cancer.
This crazy-looking chart is called an oncoprint, and it's basically the genetic fingerprint of childhood cancer. Every row is a different gene, and every single column is a different patient's tumor. It gives us this incredible bird's-eye view of the whole landscape of mutations. Now, what's really crucial is that not all those 800-plus genes are equally important. The researchers really honed in on what they called Tier 1. This is a list of just 40 genes that were mutated in at least 1% of all the samples. These are the main drivers, the big-time offenders.
And sitting right at the top of that most wanted list is this one, TP53. It has this amazing nickname, the Guardian of the Genome. Its whole job is to protect our DNA and slam the brakes on tumors before they can even start. So you can imagine, when your guardian is broken, you're in real trouble.
And you can really see just how important TP53 is here. It was mutated in 5% of all the samples. That makes it the #1 driver by a long shot. After that, you've got its key accomplices, genes like NRS, KRS, and CTNNB1. Just these 4 are responsible for a huge chunk of the problem. So what actually happens when one of these critical genes goes bad? It's not just a single broken part. No, a single mutated gene can set off this devastating domino effect, sending ripples of chaos through the cell's entire operating system.
Our cells are run by these insanely complex communication networks. Scientists call them signaling pathways. And you can see here just how tangled up they all are. They control everything: when to grow, when to divide, when to die. When a key gene in one of these gets mutated, it's like a major highway interchange gets shut down, causing total gridlock and sending signals down all the wrong roads.
An easier way to picture it is to think of dominoes all lined up. In a healthy cell, they fall in a really precise, controlled sequence. But a mutation in a gene like KRAS or TP53, well, that's like something comes in and knocks over the wrong domino, starting a chain reaction that you just can't stop. And that chain reaction is cancer. Let's look at a perfect example, the RAS pathway. This pathway is basically the cell's main grow and divide switch. Normally, that switch is really carefully controlled. It gets flipped on when it needs to, and then it gets flipped off. But mutations in genes like KRAS and NRAS, they basically jam that switch, so it's stuck in the on position permanently. And it just keeps telling the cell, grow, divide, on and on and on.
Okay, so we've found the genetic clues and we understand the chaos they cause inside the cell. But what does any of this mean for actual patients? How do we take all this amazing science and turn it into real-world cures?
And this leads us to what might be the most important question of all. We have this new most wanted list of genes. So are the diagnostic tests that doctors are using right now even looking for them? The answer, when you look at the data, is kind of shocking. The researchers checked their top-tier genes against 8 standard diagnostic panels used for pediatric cancer. And yeah, the big one, TP53, it's in all of them. Good. But look at KRAS, one of the top 3. It's only in 2 of the 8 panels. And then you get to CTNNB1, CREEBP, PIK3CA, they're not in a single one. 0. What this means is that there's a huge diagnostic gap, a blind spot. We now know about these key genetic drivers, but many of the tests being used today can't even see them. This research is basically a giant blinking sign that says we need to update our tools to match our new knowledge.
So let's just quickly recap what we've learned here. 1. Pediatric cancer isn't just a mini version of adult cancer. It's got its own unique genetic fingerprint. 2. We now have a most wanted list of the genes that are really in charge. 3. This knowledge is a roadmap to creating much more accurate diagnostic tests. And 4. It points the way to developing smarter, targeted therapies that go right after the specific weak spot in each kid's tumor.
And that leaves us with this one final, really powerful idea. As we get better and better at reading this incredibly complex genetic language, you have to wonder, could truly understanding a child's unique genetic code be the very key we need to unlock their cure?